See, today what I will be talking basically to you is extracellular matrix. See, in our body we are having cells. These are disposed of like this. Now, these cells can be thought of just like bricks of a building. Mix it, and for a big building we require some structures, also beams, where iron rods are there of different types, even bars. So these, why we are reading today extracellular matrix? The importance is that this ECM plays a very important role, I am telling you, and for our own growth, development of all cells. See, as we grow from child, from mother's womb to till death, we grow and grow, develop, age, and we can see the changes in our body. And all these changes are due to changes in the extracellular matrix. Even if we suffer from some diseases, many of the diseases are caused by changes in the extracellular matrix. Cancer also spreads only if there is a damage in the extracellular matrix. Similarly, when you will operate, you will see that the wound also heals, and in pathology you will be taught in detail about wound healing. Whether you make a surgical wound or there is some amount of damage in the tissue somehow. Maybe there is a pus formation, maybe there is an infection, maybe there is a damage due to physical assault or something like that. That entire extracellular tissue gets damaged and they get repaired. The structure, what I am telling you, what I am talking here is basically, if the cells are kept apart, so they may not be able to communicate, even if they remain as such, although physically we are seeing, they will not be able to communicate, they cannot be kept in this position also. So something has to be there to keep it fixed and to allow the cells to communicate. So there are several structures that you will gradually know. So these structures mainly composed of collagen, which is the most abundant human protein present in the human body. And it constitutes about 25 to 35%, average on an average 30%. So collagen is an ubiquitous protein which is present everywhere practically. Most important site is basically bone, skin, and tendons. So apart from collagen, there is elastin, there is fibrillin, fibronectin, laminin, and also proteoglycans, what you have said, what you have read in your carbohydrate chapter. They are all mixed in different proportions in different tissues, and they give quite characteristic histological picture of that particular tissue to perform the desired function. So if there is any abnormality of formation of any of the material, extracellular matrix, be it collagen, be it fibrillin, be it elastin, be it laminin, be it fibronectin, proteoglycans you have already read. I will discuss it tomorrow. Proteoglycans I have reserved for tomorrow. First, I will tell a few things about this less important, rather, less abundant, that is elastin, and then fibrillin, fibronectin, and laminin. Then I will discuss most of the time on collagen. And you can see from the image, if there is a damage or defective formation of extracellular tissue, you will get a picture like this. And these are basically almost incurable diseases, as lifelong diseases, except this scurvy what you are seeing here is vitamin due to D deficiency, but that is again due to defective collagen synthesis. So this can be cured by treatment. Rest of the disease cannot be cured. and it's a miserable condition, just like glycogen storage disease or whatever storage disorder we have said, there is practically no treatment. So what I have written here, shown here, is basically several fibers I have shown, and you can see in the cell also there is cytoskeleton. We are having organelle, but cell cytoplasm is just like a jelly-like material. It's not a pure solid jelly, pure hard thing. Same thing is like extracellular matrix. Extracellular matrix, all the proteins are present, proteoglycans are there, glycoproteins are present, but they are also present water. So it remains just like a gel-like substance. So in the cell, by this time you have come to know that there are many fibers. There is tubulin, there are spores, receptors. These are not cellular organelle basically. So they belong to those categories. And you see there are communicating channels also. So they not only remain present within the cell, but they communicate to the outside. And normal maintenance and health of the cell is dependent by the extracellular communication with the extracellular matrix. Some only lens I am telling you is present in the birth, from the birth, that lens mature only during cataract formation. Other tissues undergo continuous replacement and remodeling. So as I was just drawing the pictures here, see this collagen is there. Collagen is interacting with fibronectin, and integrin there is almost a transmembrane receptor is there, and it is communicating with actin. So there is a continuous connection between the cell, inside of the cell and outside of the cell. So write down the components of the extracellular are three basically. One is fibrous protein. As I was repeatedly telling, that is a fibrous protein is basically structural, and three are most important: collagen, elastin and fibrillin. And again I am telling you, it is the most abundant protein, collagen. It is a major connective tissue protein. Specialized glycoprotein is basically fibronectin, and proteoglycans I will discuss tomorrow. Already you know a little bit about it. So this is how the cells are organized. You see there is epithelial cells, there is this endothelial cell, and there is a fibroblast. And you can see the cell surfaces also. These fibers, like substances what you are seeing, these are basically constitute the extracellular components of the extracellular matrix, and they are communicating. Can you see? Without these things, the cells will not function properly. So again, just giving a schematic representation how complex is the tissue. It's not that simple as we normally see. Extracellular functions. Remember the functions, they might ask you the question, what is the function? So it's a non-cellular component of the tissues and organs. See, in our body we are having four types of tissues. One is epithelial tissue, one is muscle tissue, one is nervous tissue, fourth one is connective tissue. Connective tissue basically connects everything. Without connective tissue, no tissue will function properly. Nerve tissue will not function, epithelial tissue will not function. Muscle tissue will not also can function. So another name of extracellular matrix is also connective tissue. So they make a scaffolding type of structure on which the entire organ or the tissue they are formed. So they make a scaffold or framework, structural framework, and of course they give protection of that particular organ or the tissue. And as I told you, they play an important role in the growth and differentiation, and without that thing, the tissue or organs cannot be maintained. And I have told that through these proteins, the milieu interior and milieu exterior is also maintained, and biochemical function of any organism is dependent basically on the extracellular matrix. So you will read in your clinical classes about different inflammatory stages where this extracellular matrix will be altered, cancer will be spread, and there will be disease development. And by this time you know the all organs are unique in shape, size, and if you cut histology, that will be different also. Because each organ performs different function. The function of eye is quite different than that of function of kidney. So they must not think that the ECM component of eye will be same as that of kidney or a bone. So one important point, as I was saying, okay, water. It's a gel-like substance. These gel-like substances, because in water these substances are basically present. So this I have told. The form of elastin. Just one point. I have shown you. If you see the ordinary microscopy, you will see the elastin fiber like this. You will not be able to identify other than unless you make a special staining. So elastin is formed from tropoelastin. Tropoelastin is having two fates, with cross-link or without cross-link. If it is without cross-linking, that is an immature tropoelastin, that is not functional. Mature elastin is a cross-linked elastin, and this is accomplished by post-translational modification, and there is an enzyme called elastin which breaks down, and again these cross-links are broken down. So elastin becomes non-functional. One important point you have to remember, that elastin is present when the tissue is stretchy, some spring-like action must be there. So elastin, the name has come, it must have elastic-like property. Blood vessels, with each systolic contraction, basically contraction, there will be increased expansion of the vessel wall. Wherever there is extension and recoiling, these tissues are present, and that's why they are present in lung, urinary bladder, uterus, large blood vessels, some ligaments. When we stretch or joints, so they get stretched. So they are tightly associated with fibrils. 1/3 residue is present in glycine. This important point is desmosine link, cross links. You see here, this is quite characteristics of elastin, not present in other tissues. Note down this point only. Mature elastin, desmosine like cross links are there, and there are 4 lysine residues are here. So this part is very unique, that you have to remember. The differentiating point between the collagen and elastin is there is no glycine-X-Y sequence, no hydroxylation, no glycosylated hydroxylation, and triple helix structure also. This I will tell you in detail in a tabular form. So these are the characteristic features of elastin. আরেকটা আছে দেখো তো, আগেটা যাও, এটা কাজ করছে না। হ্যাঁ ঠিক আছে, ওকে। Next point is fibrillin. It's a major structural protein of microfibrils, and it is secreted from fibroblast. So fibrillin, most important point here is, if there is a defective fibrillin formation, it causes a disease. Its name is Marfan syndrome. Fibronectin. Another important point, you see here the Y-colored substance. This is fibronectin. These fibronectins are present. This is epithelium, basement membrane, and it is in continuity with the interstitial matrix. There is a gap. It has been written schematically just to show you that these fibers are communicating. The basement membrane is communicating with this interstitial matrix, and these fibers are also communicating with the cells. What are the fibronectin binds with collagen. You see there are fibronectin, there are 3 motifs. One is 1, 2, and 3 types of motifs are there. There are 4 places where it binds with collagen, fibrin, heparin, and there is another site for cell also. So with this collagen, this fibronectin binds with these cells, and it is having 2 strands. This is a two identical subunits, it is a dimer, and there is a two disulfide linkage is also here. This is characteristic feature of fibronectin, and this is insoluble in network, and this fibronectin is important for wound healing. So you have to give some medication or you have to apply something which will help the formation of fibronectin so that the wound gets healed as fast as possible. So you see here this fibronectin, this is fibronectin, this is alpha and beta chain polypeptide chain of integrin. Integrin is basically a transmembrane protein. So transmembrane receptor is there, along with fibronectin is working, another side laminin is there. So here through this integrin receptor, the fibronectin is reacting with collagen, here interacting collagen outside, and through four attachment proteins, this is communicating with actin. So all these structures are connected to each other, and that is the reason why the cell is kept in position. Otherwise, the cell will get detached. So can you see the tight linkage between the components from actin through four attachment proteins through integrin receptor, that is a transmembrane protein which is connected with fibronectin, that is connected with collagen. So this is the complexity of the structure, and that's how the cells also function. Laminin. Laminin is again a major component of basal lamina. You see there are also places it is having three chains, one alpha, beta and gamma. This is biochemistry part you have to remember only. So laminin is attached to the epithelial cells, and it influences cell differentiation, migration, adhesion, and all these extracellular matrix proteins are involved like this. So lens, if you do the lens confocal microscopy, you find corneal cells are like this, but laminin is shown here also. In cornea, collagen tissue is present also in abundance. The cornea is transparent. One important point of glomerular filtration. All of you know that albumin cannot pass through the glomerulus because of the large size, which is indeed not so. Because you see there are pores are here, the slits through which the glomerular filtration takes place. So there are two separate cells. One is forming from the Bowman's capsule side, another side is blood side, that is capillary side. It allows small molecules like inulin clearance test you have read, because inulin can pass through. It's a very small molecular weight protein, 5.2 kilodalton, but not large proteins like plasma proteins. But pores in the glomerular membrane is about 8 millimeter across. So more than this cannot pass, ideally. So albumin is a diameter of 3.8, ideally then albumin should pass. Why it should not pass? Because the pore size is quite half of the size limit, but only thing is that this is prevented by the negative charge of heparan sulfate and sialic acid containing glycoprotein present on the basement membrane. So normally albumin, you remember that pK value is 3.1 or 4.1, 4.7. So albumin remains negatively charged with the normal body pH. So there is a repulsion by the heparan sulfate, negative charge of heparan sulfate and sialic acid at the glomerular basement membrane, and that is the reason why albumin cannot pass through the glomerulus. If there is only disease, then only it will pass through. Otherwise not. So disease must damage this thing, that structure, then only it will pass through. Next, what are the keratin? Keratin you all know. Keratin is present in the skin, hair, nail, and it is composed of alpha keratin, keratohyalin. I am not going into the detail. You read it from the books. Major component, just write at least these five points. Elastin. Elastin is present in only tissues requiring stretchiness, like lungs, bladder, blood vessels. At least one line you must be able to tell. Fibrillin. It is present in microfibrils, both in elastic tissue and non-elastic tissue. Fibrating, as I have explained, that it is anchoring the cells to the ECM. A single function of at least one protein, that's why I have written. Laminin is the major component of basal lamina, and keratin is present in our hair, skin and nail. At least out of these 3, 4, you must be able to tell these 4 things. What are the major components? Elastin, remember, again, this tissue requiring stretchiness, elastin, elastic. Fibrillin is for microfibrillin. Fibronectin, I have repeatedly telling it is anchoring the cell to the extracellular matrix and keeping the cell fixed. Collagen, I will discuss in detail about collagen. So, collagen, you know, must know everything, because this might come as a long question, this might come as a short question. These are the things which so far I have told, may come as a short note. But collagen, try to hear from me as much as you can, and then you simply read it. So, it is a major connective tissue. I have been repeatedly telling you the major, most abundant protein of the body. Till the previous edition book, there are only 28 types. The recent edition book only says 29 types of collagen are identified, although there are 30 genes. So, in human beings, type 1 and type 2 is present mostly. So, they confer rigidity, they confer stability. Normally, it is present in the triple helical structure, and usually there is 2 identical chain and 1 slightly non-identical chain that is differing in composition, amino acid composition. So, they are held together in a triple helical form. So, 29 types of collagens are present. Most of the collagens are secreted by the fibroblast. Where from collagen is secreted? Fibroblast. A video to dekho. Ei dekho fibroblastic structure. See, these collagens are formed. This I will tell stepwise. বন্ধ করো কয়টা যাও See, there are 28 types. It is very difficult to remember each and every type. At least either you remember the first 4 type, or at the best type up to type 5. So, type 1 you can remember as scab. You can write type 1 is basically present in the skeleton. Remember, it is most important is bone. There are 2 ways how you can remember it. Type 1 is present basically bone, skin and tendon. This is the main component. So, BST, that is how I used to remember. So, bone, skin, tendon. Type 2 is present in the cartilage. Type 3 is present in the reticular fibers, main component. Skin, lung vessel, and type 4 is basement membrane. So, you can remember scab just like that. You can otherwise skeleton, cartilage, artery and basal membrane, basement membrane. And there is one mnemonic I have seen. So, be so totally cool. Read books means all the important structures which are formed by these collagen fibers will be present. So, there are mainly 5 types. You remember, if you cannot remember, then at least there are 4 types, and type 5 is basically type 4 basement membrane or basal lamina. What are the functions of collagen? Write these 4 points. So, it is secreted by the fibroblast. I have shown you. This undergo extensive post-translational modification And this post-translational modification you have to remember for examination purposes. So this I will discuss. By this time, at least functions of the collagen. What are the functions? It's basically, as I told you, it's basically making the structure. So it is giving support to the organs and providing tensile strength and regulation of cell adhesion, alignment of cells for anchoring, that I have clarified. Support chemotaxis and migration. See, there must be some guideline, there must be some path through which it will travel. So again, direct tissue development helps in proliferation and differentiation. These are the functions of collagen, whatever you remember, either individually or collectively. So in blood vessels, when if there is any damage in the endothelial cells, the collagen tissue gets exposed and that initiates coagulation. So that you must remember. This triple helical structure of collagen is important. See, triple helical DNA is a double helical structure. Remember, collagen is a triple helical structure. And there is an absolute requirement that it must be a glycine-X-Y position. These three tripeptide repeats will be there. So glycine has to be present because glycine is the smallest amino acid, and that smallest amino acid, that size is very much important to be accommodated within the space available. And X position is usually proline and Y position it is usually hydroxyproline because they confer rigidity. So a rigid structure must be there, otherwise it will not be able to withstand the other pressures. So normally it is glycine X, first position is always glycine. So glycine-X-Y means every third amino acid will be glycine. So 1/3 content of collagen will be glycine. And next will be proline and hydroxyproline. Instead of that hydroxyproline, at times you can get also lysine. So lysine or hydroxylation. So this entire polypeptide chain is first undergoing left-handed helix. Then the three polypeptide chains undergo right-handed helix. So that is the basic formula, basically how the collagen is formed. And then once this is formed, they are arranged in a quarter staggered alignment. Remember, this is characteristic feature of collagen only. Quarter staggered attachment. Quarter means 1/4 of particular collagen fiber which is above or which is next to that collagen will be absent. I can show you the picture. Next to পরে দেখো তো পরের পিকচারটা দেখো তারপর আসছে এটা একটা আগে যাও আমি পরের পিকচারটা দেখাচ্ছি ওকে ফাংশনস কো এগো হ্যাঁ নেক্সট proline and hydroxyproline confer rigidity. Why it is required? একটু বড় হয়ে যাবে Glycine's smallest amino acid to be accommodated in the space, and the post-translational modification is most important part. Proline is made by the prolyl hydroxylase, is formed hydroxyproline, and lysine at Y position is hydroxylate called hydroxylation. And if there is a defect in the prolyl hydroxylase or lysyl hydroxylase, you will get the disease. So collagen fibers are stabilized by the covalent bonds, and that gives a tensile strength. So collagen is first formed like a pre-pro-collagen, just like insulin. Then pro-collagen is formed. This pro-collagen then is expelled from the Golgi apparatus. Then it undergoes tropocollagen and this collagen matrix, and then it undergoes a quarter staggered arrangement, and then further stabilization is made by covalent interaction between intra and interchain by lysyl oxidase. And this is quarter staggered arrangement. What I want to say, so this is the structure. This is the structure of a particular triple helical structure. You see the triple helical structure is there, and only 1/4 it has come and stopped here. So it has come and stopped here. A little, it is not exactly above. So this is the arrangement which is giving a bead-like appearance. So this you must remember, this quarter structure arrangement of collagen fiber, and that gives a very good packing. So cross-link basically two collagen, two triple helical molecular interconnected with each other through aldol condensation, and that is what is called cross-linking. That cross-linking is important. Without cross-linking, it will not remain stabilized. So bundles will not remain in a stable position. At least after not a step, if you cannot remember at least whatever underline, you must remember this thing. This might come as a short note. This has been told repeatedly to tell you. Intracellularly, there is a leader sequence. So first, intracellularly, that cleavage of the signal peptide is removed first. Then proline are hydroxylated by hydroxylation, and this step is very important for the collagen stabilization, and some of the hydroxylation residues are also glycosylated, and the formation of intra and intra-chain disulfide bond to make it stable, and then the formation of triple helix. Up to this part is intracellular, and then extracellularly there are some excess amount of peptides which are present both at the amino terminal and carboxyl terminal. These are cleaved. That is number one part. Then that arrangement of quarter staggered arrangement, what I have shown you, it is done extracellularly, not intracellularly, because it is forming the extracellular matrix. Remember, it is not intracellular. So that structure is made extracellularly because such a large structure cannot come out from the cell also. Then there is oxidative deamination of amino acid and lysyl hydroxylation, as I have already shown, and there is intrachain and intrachain cross-links by skip base and aldol condensation. What is that? So normally collagen can remain in a fibrillar form or as a non-fibrillar form. So only 5 types are present in the fibrillar form. The rest of the collagens are present in the fibrillar form, non-fibrillar form. What is that? A difference just note down. Difference collagen and elastin, what are the difference? All 5 points are no with elastin. See, triple helix structure is present in collagen, not in elastin. GLY repeating sequence is there, no GLY sequence. Hydroxylation is present, no hydroxylation. Carbohydrate containing, no carbohydrate. Presence of extension peptide, no presence of extension peptide. This might come as a short, this MCQ question also. Which of the following is not present in elastin or which is not present in collagen? And many different genetic types, as I have shown you, there are 29 different types, so there are 30 genes. But elastin is made from one genetic type. So one gene is there. And intramolecular aldol cross-link is there. You see here, this is intramolecular, this is intramolecular desmosine. During elastin, I categorically told you, remember one part, that desmosine and that 4 lysine, these are quite characteristic. So desmosine like cross-links are present in what? In elastin, collagen, fibronectin, something like that. That's my question. Now, what are the uses? দেখতে পাচ্ছ নেটওয়ার্কটা কিভাবে তৈরি হয়েছে? This is quite different. এবার দেখো ওয়াইন্ডিং যেটা দেখাচ্ছিলাম, যেভাবে বললাম প্রথমে লেফট হ্যান্ডেড তারপর রাইট হ্যান্ডেড, দেখো কীভাবে ঘুরছে দেখো। So first left handed, then right handed. সেখানে আনওয়াইন্ডিং হলো, জাস্ট রিভার্স করলাম। যে ফরমেশন হয়েছে সেভাবে স্টেপ ওয়াইজ ঠিক উল্টো দিক থেকে ভেবে type 2. Out of many videos, I thought that this will be most important. These I have taken from here only. So they have said 4. So 90% present in the bone. Defective product type 1 collagen. কম্প্রেসর শক অ্যাবজর্বারে কাজ করে, লুব্রিকেন্ট হিসেবে কাজ করে, যেখানে হাইলোনিক অ্যাসিড সেখানে লুব্রিকেন্ট, এখানে শক। This is a peculiar thing. Many things, many persons will be able to see that they can bend their fingers like this. So immune-mediated damage is there. You see, the basement membrane, there is auto-antibody. So that membrane is damaged, means gone. That's what I told you. One disease also, autoimmune disease, they are also called. So collagen synthesis, you can see GLY, GXY, that I told you, that you have to remember there is a unique sequence. 1/3 is always glycine, cholinolysine. So important point is, as vitamin C is very much essential, and copper also. So in case of wound, you can give vitamin C. That will help rapid recovery. যেটা দেখাচ্ছিলাম, প্রো কোলাজেন টু কোলাজেন, দুটো মাঝে টার্মিনাল কেটে দেওয়া হলো, carboxy terminal. Lysyl oxidase is important enzyme that I am telling you. Copper is important thing, that is for cross-linking. A defective protein or defective cross-linking of collagen molecule, and that will give rise to disease. ये देखा तो ताड़ाताड़ी ये प्लस ताकि छेड़ दो चार्टर में जाओ उधर ढूंढ के दो अब disease due to abnormality. Ehlers-Danlos syndrome to আমরা when we were student we knew only there is one type, but there are at least 4 different types of collagens involved, and there are 13 subtypes as per the latest classification. এই শুধু এই চারটা ভালো করে পড়ে নেবে, মনে রাখবে। জাস্ট রিমেম্বার osteogenesis imperfecta, what is the defect? I have written here, but you remember like Ehlers-Danlos syndrome. What is the defect? Defect in lysyl oxidase and hydroxylation, that is not allowing the collagen to form, cross-link. So hyperextensibility, many persons may not have a complete type. There are 13 different types. So as I told you again, we can many persons again you can see they can bend like this, they can bend more than what we can do. So there is some defect in the that earlier it was called cutis hyperelastica. So Marfan syndrome is again there. fibrillin 1 gene. This is a common question. These are the two common questions I am telling you. Osteogenesis imperfecta, what is the common cause? What is the cause? So here basically why this is so defective, because glycine is present. This glycine is most important thing, that is replaced by serine residue. Menkes disease, again defective cross-linking. Ascorbic acid deficiency, again there is defective cross-linking. Hydroxylation, prolyl hydroxylation. Homocystinuria you have already read, again that causes blocking of the cross-linking. So again osteolateralization. You make one chart, I am telling you, just to remember and somehow remember before your examination. And this will be again required during your NEET PG also. Usually from the storage disorders, from the uncommon diseases, usually one question is there as an MCQ. That you must remember and you have to somehow remember this thing. So I have read it, written it. You can, I will make a small chart and you can read also. Epidermis bullosa, one child I have seen, I do not know in the OPD, whether it was a case of epidermis bullosa or it was a pemphigus, bullous pemphigoid, I could not identify, but that was a very severe case. That picture is with me. Aliport syndrome, that you have seen that there is a damage in the basement membrane and it can cause end-stage renal disease. What are the hard dive? So, in summary is basically collagen is the heart of all ECM, is the heart of extracellular matrix in all tissues. There are 29 different types, most abundant protein, and then you have to remember elastin present in the, where there is elastic recoiling of the tissues, it just acts like a rubber band. Lungs, urinary bladder, blood vessels, these are present. Fibrillin is present in the microtubule and its absence causes, defective gene causes Marfan syndrome. So, if there is any defective gene in the collagen formation or oxidation or cross-linking, that will give rise to, disorders of the extracellular matrix. Tomorrow in the first part we will try to finish this glycoprotein part or proteoglycan part.